Ever since 2020, I have been working in a wet lab, but I am quitting for a dry lab. Hi, I am Isaac. If you're new to this channel, you are welcome. On this channel, we do talk anything related to statistics or genomics analysis. I have a master's degree in bioinformatics and an undergraduate degree in biotechnology. I am a self-taught data analyst and I have helped and trained hundreds of students in statistical analysis, programming, proposal writing, report writing, abstract writing, and all sorts of academic work. And as of now, I am in the verge of quitting my wet lab job for a dry lab environment. Let me tell you why I am leaving this field. Back in my undergraduate program, that is biotechnology, we did a lot of molecular biology. We did sort of DNA extraction, RNA extraction, PCR, sequencing, and all sort of these molecular work. And it was very interesting. It was amazing. In fact, it was something that I really had much interest in. But when I was doing a job search, I did not find work in the molecular biology field. Rather, I found work in a clinical microbiology setting. So I started working in clinical microbiology around February 2020. Everything has been going on well. I was working in a biosafety level 3 laboratory. Even when COVID-19 hit, our lab participated in the testing of the COVID-19 patients because the main reference lab that these testings were being done was overwhelmed. I participated in it. I was so proud because now I was making impact to the real world. This was no longer school work. This was real work trying to impact life in this world, and I was so happy. But deep down, I wanted more. I knew that was not my environment. It wasn't a kind of setting that I wanted to be there long term. It was just a temporary solution because when you leave school, you want to get something to do. You want to get a source of living. So in 2022, while working still within the lab, I started my master's program in bioinformatics at Makerere University, and things started shifting in the way I wanted because now I was going deep into my genomics work, my molecular work, things like sequence analysis, writing computer script or automating tasks using machine learning, some of this cutting edge technology which is coming out currently. So I've been feeling at home ever since then. Even my master's project, I used machine learning to predict internalizing mental disorders amongst children and adolescents with HIV. It wasn't easy at first, I must confess, but the experience was worth it. So when I defended my final thesis in November 2024, I knew I had to make the bold step. One thing that I am so grateful about while at school were my supervisors. My academic supervisors were so encouraging. They advised me to begin sharing my work at different scientific conferences, have talk with other researchers, or joined a working research group. And that is what I exactly did. As I talk now, I applied to over 6 different scientific conferences and all of them accepted me. This is strange to say, but I've not had any rejection from my abstracts that I submitted to different scientific conferences. And if you want me to make more details, how I designed my abstract, how I designed my posters for presentation, how everything has been going, and I teach you all the lessons about what I went through, let me know within the comment section. Then I will make a dedicated video talking about abstract preparation, poster preparation, presentation preparation, and then you also prepare yourself. The more I have been attending these conferences, the more I have shared my work with the people, and I have gotten very constructive feedback from someone giving me a new method that I could have used for my analysis, to asking me some questions that I wasn't thinking about within my work even itself. So more and more, I have been feeling at home. This is my environment. This is my family. And I have realized that this dry lab path can really open many doors. Then I've received so many comments from you people in one of my videos talking about don't do bioinformatics, and so many of you have asked me questions, wanting advice, and I hope this video is partially answering those questions you have been having. As I sit here, I've realized that I can help so much people. I could build tools, I could automate analysis, I could participate in cutting edge science. The genomics field is not saturated yet. There are so many things to be learned right now. I want to go and explore those new environments. Don't get me wrong, I have learned so much from the wet lab. I've had a very strong foundation, and I think it is fairly important for anyone who wants to go into a dry lab setting, data science or bioinformatics, to a little bit also interest themselves with the wet lab. Let me give you an example. In the microbiology lab where I have been, one of the tests I used to do was drug susceptibility testing for tuberculosis. Now, this was a phenotypic test. You set an experiment without any anti-TB drug, and then you set others with anti-TB drug, and then you monitor their growth for some time. If no growth occurs, then that sample is sensitive to that particular drug. If you see growth occurring, then that could mean resistance to that particular drug. But whereas that one may tell you something, it may not also be 100% accurate. What if that sample, those organisms were sequenced? Then I performed whole genome sequence analysis. I look for the genes responsible for drug resistance. If that sample has that gene, then I can confirm that yes, what I saw phenotypically truly means that that organism was resistant. If I don't see the resistant gene at the genetic level based on my dry lab analysis, then I cannot trust the wet lab analysis. I am not saying this is black and white, wet lab and dry lab, but all I'm saying is that these can all be mixed together. One can aid another. The major challenge that is forcing me to truly move away from my wet lab setting, it's because most of the labs are rigid. They are not open to change. They're not open to adjustment. If you suggest that, I think let us add an additional test here. After a sample becoming positive, let us do sequencing and then we do analysis. Then we can report both the phenotypic result and then the genotypic result. But most people who are purely wet lab based, they are not open to such ideas. Their assumption is that you cannot trust computers to give you reliable results. And I understand the perception because this is translational science. And I know things like bioinformatics, genomic analysis is still highly doubted by some community, but one step after another, we will get there. Of course, I am not forgetting the freedom of where to work, when to work, and how to work. I love freedom. I would prefer to be working from home, working from anywhere, working in my farm deep in the village, or just from anywhere I want. It's something that I am interested in and I've not yet been able to achieve. So I hope that if I transition fully, then I may be able to achieve that. But most importantly is my future. When I look into the next 3 to 5 years of my career, I don't see myself in a wet lab setting. I see myself in a dry lab setting doing PhD or postdoc. And I want to do bioinformatics in that line. So it's only fair that I move as soon as possible so that I learn as much skills as possible so that when these offers come, I am ready to take it heads on. Currently, I am working with a research group. This research group mentors students. They give you projects, you analyze, you write report, you write manuscript, you do presentations, and then you build your muscle around bioinformatics or dry lab in general. And I want to give it most of my energy. I want to spend this time really doing what I should be doing for my future career. I know the shift is not going to be easy because I am leaving a paying job for something which is not yet right now paying. But I think it is a sacrifice worth taking. Based on the emails that some of you do send me and the comments I do receive on some of the videos, some of you keep wondering, how can I make a switch from my wet lab setting or from my career that I am doing right now to bioinformatics or data science in general? You keep on asking yourself, is it something which is easy to do? Is it something which is worth it? Or if I want to do, how can I do it? And well, I can help you, right? I believe I can, since I have walked the journey. I know what is most important. I know what you should concentrate on most. At least I have that experience based on what I went through and the different students I have been working with. And in case you need that assistance, well and good, we can jump on a call together. I'm going to assess all your needs, all the current skills you have. Then I will design a very well personalized training just for you. This field is a very high skilled specialized area, right? It's a bit hard to do everything together with everyone. The field is very wide. Metagenomics, human genetics, plant genetics, RNA, epigenomics. There are so many areas that everybody needs their own personal plan. So if you're ready and you're serious and you want a clear pathway to start over, you can work with me one-on-one. Send me an email or book a one-on-one consultation using the link below in the description, and we will have a talk, me and you. Otherwise, let me know, which team are you? Are you team dry lab or are you team wet lab? Let me know within the comment section. And if this video has been helpful, like and subscribe. Share this video with someone who may find value in it. Watch this video next if you're serious about data analytics in general.